Okay. Hello, my name is Alexei Lopez, and today I'm going to talk about the role of proteins in adhesion. This is part of group 2. Okay, we're going to start with the overview of the general information. This has to do with the surface carbohydrates on the cell serve as points of attachment for other cells. Infectious bacteria and viruses, toxins and hormones, and many other molecules. The ability depends on the presence of sugars, the ability and accessibility, and their mode of presentation. The carbohydrates recognized by the cell adhesion molecules, which are glycoproteins expressed on the cell surface. Several viruses, bacteria, and parasites have exploited the presence of cell surface oligosaccharides, using them to enter in the cells. Rhinoviruses attach to the ICAM-1, which is the intracellular adhesion molecule -1. Parvoviruses attach to the erythrocyte-specific cell surface P anti-influenza virus. The Plasmodium falciparum. So it's a falciparum is protozoan causing malaria, using glycophorin, the MN blood group system, to enter the erythrocytes. The herpes virus type 1 enters the cell via the FGFR, which is the fibroblast growth factor interceptor. Okay, now let's talk about the structures and the various proteins involved. As we can see in the picture on the right, it describes, for example, the ABO blood group. Antigens are the carbohydrate moieties attached to the lipids or proteins on the surface of the and the erythrocytes and many other cell types. The A, B, and O antigens are structurally related. All people have enzymes, or in other words, proteins, which synthesize the O antigen, which is the H substance in people with B antigen. There's an enzyme which adds galactose residue to the H substance, and people with A antigen have an enzyme adding to the GalNAc residue. Okay, location in specific organisms. Such example includes the trypanosoma brucae. It causes dangerous diseases within some animals, which contains highly variable surface glycoproteins, or VCGs. They are strong antigens. Trypanosoma frequently changes one VCG to another, thus enabling the protozoan to elude host responses that would inactivate it. And as stated earlier, the other example can be Trypanosoma brucae. It causes dangerous diseases within some animals, which contains highly variable surface VCGs. And this is one disease that could be related to the recognition and adhesion. And the recent finding about And the recent finding is that the vessel wall through the recognition of specific carbohydrate ligands, which often contain sialylated, glucosylated, and lactosamines, such as sialyl and Lewis, in the excellent gene expressed by activated endothelium, which has been shown to support the adhesion of the S-like sphere in colon cancer cells. In the present study, we examined the interactions of multiple colon cancer cell lines in all three selections. The three colon cancer cell lines, which are actually mentioned, which is the LS180, T84, and the COLO205, bound to the recombinant purified E minus, P minus, and the L line selection. And the questions that I have about my topic today is that what truly major role does the environmental factors play into the role of glycoproteins in adhesion, and how much would the control group differ from the actual population if a test rule about role of glycoproteins in adhesion was established? And this is officially the end of my project. Thank you for listening to me.